We're going to go ahead and jump in today. So welcome everyone. Thank you for joining us for this webinar with Wave Life Sciences. I'm Eric Camino. I'm PPMD's Vice President of Research and Clinical Innovation. I am joined today by Paul Bolno, the President and CEO of Wave, and Anne-Marie Lee Kwai Chung, Wave's Chief Development Officer. And they're joining us today to talk about some interim results from their open-label phase 2 study, FORWARD-53, which is studying their investigational product, WVE-N531, which is an exon skipping product for patients amenable to exon 53 skipping. So they are going to walk us through a presentation. At the end of this, we will have an open Q&A. So if you have questions at any point during the presentation, there's a chat function at the bottom. Throw your questions into there. We will try to get to as many of those as we can when we get to the Q&A at the end. We're also recording this webinar, so if you have friends or family that this would be relevant to, it should be posted up on PPMD's website within the next week, and we can share that with them. And if you're watching on the recording and you had questions that maybe weren't answered during our Q&A process, please reach out to PPMD. We'll make sure that we can get you in touch with the folks at Wave and get answers to those questions. So with that, I think we can get started this morning, so I'm going to hand it over to you, Paul.
Thank you, Eric, and thank you all for joining us. Good morning. A decade ago, we recognized an opportunity to apply our novel oligonucleotide platform to advance therapeutic options for Duchenne muscular dystrophy. And I can say this with a lot of sincerity that over the years, our culture and our commitment to always follow the science has been directly shaped by the resiliency of the DMD community. I often reflect on a conversation that I had a number of years ago with the unfortunate setback of suvodirsen, and a call that I had with Pat the night before we announced that data. And the conversation we had together was that Wave remained committed to the DMD community and again would continue to find a path forward. When we go into the work that we've done subsequent to the suvodirsen data and really dug into the science as to why that didn't work and what we would need to solve for in order to advance the therapy that could transform the treatment of DMD, we spent a lot of time focused on chemistry, models, and that led to the data that we'll present later today. Charlie, if you can go to the next slide. The work that we did on preclinical models for N531 was important. We fundamentally advanced chemistry, so using what's called our PN or phosphoric guanidine. This is a new backbone modification that really opened up delivery to muscle cells, heart cells, and diaphragm. So things that involve not just skeletal muscle and ambulation, but potentially cardiac function and respiratory function. We studied N531 in two important preclinical models. The first preclinical model was the double knockout mouse. This was an important area of exploration because in this model, there's no dystrophin and there's no utrophin. So importantly, the functional changes that we can measure and explore in this mouse are directly related to the production of dystrophin. This is an exon 23 amenable mouse. So this is chemistry that's applied to a backbone surrogate for study, but it does give us an opportunity to look at chemistry and distribution in the model. What we saw remarkably was an increase in dystrophin expression in skeletal muscle, but even more impressive was that we had higher levels of dystrophin in the heart and diaphragm than skeletal muscle, meaning as you hear about data later, that we may be underrepresenting the expression of protein in two important areas. Moving from the mouse, and again, before human clinical studies, we went and looked at the non-human primate. And this gave us an example of being able to look at this translation of our new chemistry in now non-human primates, where we could look at exposure and translation of where the medicine was going in not just the skeletal muscle, but heart diaphragm. Again, we saw substantially higher concentrations of N531 in the heart and diaphragm. So this consistency of data, this consistency of work and exploration that we have on this backbone was able to demonstrate that we could see that we were getting these levels in important tissues. So as we think forward about potential therapies and the design for N531, obviously there's a focus on the current clinical study that's exploring in ambulatory boys. But as we think more broadly about the applicability of this program being able to get exposure to heart and diaphragm, we think is going to be important in the non-ambulatory setting. And again, being able to think about, and we'll share more data that we have both preclinically, but Anne-Marie will share clinical data, that we do see expression in getting medicine into the stem cells in the muscle, so the regenerative cells. So really being able to think about early treatments, middle treatments in terms of ambulation, but also being able to explore beyond that into the non-ambulatory setting. And so I know what people are here for. And so with no further ado, I want to turn it over to Anne-Marie to walk through the clinical data and how this translates.
Thank you, Paul. It's a pleasure to be with you here today. I'm going to start by just explaining a little bit about the ongoing FORWARD-53 study, and then take you through the results that we have, and talk a little bit about what's going to happen from here. So without any further ado, let me start by explaining FORWARD-53 is our ongoing, potentially registrational study of N531, the phase 2 study in boys with DMD amenable to exon 53 skipping. And the key endpoints of this study include safety and tolerability, pharmacokinetics and pharmacodynamics, including exon skipping and, of course, dystrophin expression, measured after 24 and 48 weeks of treatment. The functional measures that we are assessing, we assess at baseline and, of course, week 24 and 48 weeks. Now, the patients in our study are earlier in their disease course, and for that reason, the progression that you would have of the disease in this study is thankfully slow. So at 6 months, you wouldn't expect to have seen a significant amount of disease progression for us to be able to measure what any impact of N531 would have on that disease progression. But that is something that we're looking at throughout, and it's something, of course, that will be important to look at week 48 of the study. So this is an interim analysis after 24 weeks of treatment, and we've done a pre-specified analysis of dystrophin protein in all of the boys, and also an analysis looking at the ambulatory boys. In terms of baseline disease characteristics, there are 11 patients in the study with age range 5 to 11 years, and they are on a stable steroid regimen. The boys in the study, 10 are ambulatory, 1 boy is non-ambulatory. We opened the study to non-ambulatory boys who had not lost their ability to ambulate within a year of the start of the study. And there are a number of different underlying mutations, but they're largely occurring in 45 to 52 region. The safety and tolerability of N531 at this interim analysis was really very encouraging. So all the treatment-related adverse events were mild. There were no serious adverse events, there were no discontinuations, and we saw no oligonucleotide class effects. We know that safety overall is very important for boys with DMD and their families, of course, and this is something that we'll continue to collect as we run through the study, and we'll report out at the end of the study. So moving to the dystrophin analysis. So we saw a highly consistent expression of dystrophin across the boys, with dystrophin reaching up to 14% of normal in a muscle content adjusted measurement. We took biopsies from all 11 boys. 1 biopsy was found to be too low quality to be analysed. And as I mentioned, there was 1 non-ambulatory patient. And knowing that patients who have lost ambulation may not have the same response to drug, we pre-specified an analysis to look at all, but also the ambulatory patients. So the results that I'm reporting here, the mean absolute dystrophin levels in the ambulatory patients was 9% in the muscle content adjusted measurement, and 5.5% without muscle content adjustment. We saw 2 isoforms of dystrophin that are consistent with Becker muscular dystrophy phenotype, and consistent with milder disease progression in that phenotype. So as I mentioned, this consistency is something that's important because obviously every individual patient wants to have the best response possible, but also when we think about how we will design future studies, knowing that there's a high level of consistency between patients gives a greater ability in the confirmatory studies to show that dystrophin is translating through to functional outcomes. So this is something that we were really encouraged to see. 89% of the ambulatory boys had levels exceeding 5%. And yeah, something really encouraging as we move through the rest of this study. So moving on to talk about what else we saw in the study. So in terms of the muscle concentration, we saw really high muscle concentration from the muscle tissues, 41,000 nanograms per gram. This is consistent with what we saw in our Part A clinical trial. This part of the study looked at 3 boys who had been treated with N531 for 6 weeks. The tissue half-life is now estimated to be 61 days, and that's supportive of us moving to a monthly dosing interval. And that's also something that we think is going to be really meaningful for the boys and for their families moving forwards, both in this study, because we will be moving our patients in this study onto a monthly dosing regimen, and also hopefully in future when the drug eventually, we hope, will be with more patients in a marketed setting. We also observed a high level of exon skipping with a mean of 57%, which means that we have, with N531, restored the reading frame for dystrophin. So to achieve exon skipping, N531 has to access the nuclei of the muscle cells. And we have confirmed that with this interim analysis. So on the left, you can see that we are showing that N531 is present in the myofiber nuclei. That's shown by red staining. And on the right, we're showing that N531 is being absorbed into the myogenic stem cells in the majority of boys. This is just one image, of course. And on this image on the right, you can see using PAX7 staining that's brown, we can see that N531 is in the stem cells. Now that's important because stem cells in DMD, in the absence of dystrophin, they're not able to undergo the division that they need to regenerate the muscle cells, and that means they are diminished in their ability to repair. And we believe in restoring dystrophin in muscle stem cells will help them function as they should and undergo the division that they need to preserve their ability to renew and to initiate the repair of the muscle fibers. And so the drug getting into these important cells could be something very important in future, so that we can go on to demonstrate these changes in muscle fibers results in functional benefits for boys. We did not take baseline biopsies in our study, and we did that because we're very conscious of the huge impact that has on patients. There's really extensive data available around exon 53 PPMD and the amount of dystrophin patients we've had at baseline. So we took biopsies at week 24 and will take another biopsy at week 48 of the study. But patients who completed Part A, who rolled over into the FORWARD-53 study, we have 2 biopsies that we can now compare. And so I'm showing here an example of one of those boys, which we think is helpful in understanding what's going on in the study overall. So on the left, you can see data from a patient from Part A, and on the right, their equivalent biopsy from the 24-week time point. And what we can see is that we are seeing evidence of myocyte regeneration by the increased number of internalized nuclei. That's marked in black arrows. So you can see moving from left to right, the number of black arrows is increasing. And we're also seeing an improvement in general in the muscle health and things like the regularity of the myocytes, and you can see that in the bottom row moving from left to right. And these results are very encouraging because it's understood in general that you couldn't see these kinds of improvement in the muscle health without functional dystrophin being present. So over the course of the study, we're looking at serum biomarkers that could be indicative of changes occurring. And we saw clinically meaningful decreases of these biomarkers here, CK and AST, that are indicative of muscle health. And the CK was significantly decreased from 12,000 units per litre to 6,000 units per litre. That's the panel on the left. And similarly, AST decreased from 300 units per litre to around 200 units per litre. And these are both clinically meaningful changes. And CK does change when patients have steroids introduced. And the magnitude of change we're seeing here so far in this study is greater than what you would see with the introduction of steroids. And these patients are already on stable steroids throughout the study. So this is something that I think we found extremely encouraging. Of course, we need to complete the study and complete our analysis the whole way through 48 weeks. But in the context of having seen dystrophin, this is something that we found exciting. So in summary, at this 26-week interim, we see a really consistent amount of dystrophin being produced across patients. And we see that the drug is in the right place. There's a really significant amount of drug in the muscle where it needs to be to have its effect within the cells and the nucleus. The half-life is such that we will be able to move to monthly dosing, something we think that is going to be very helpful for boys and their families. We've had this preclinical data that suggests that N531 actually can have potentially greater effects in the heart and diaphragm than it has in the skeletal muscles. And so obviously future study of this is going to be important to better understand the benefits that N531 could have. We're very encouraged by what we're seeing in the biomarkers, the serum biomarkers, and also what we're seeing in the actual tissue. So looking at the stem cells, looking at the actual histology, and seeing that muscle fibers do appear to be regenerating. And we're very encouraged also by the safety profile, having not seen SAEs, discontinuations, or class-related effects. This whole study, the whole program, would not be possible without support from the DMD community, and I think they provided us some really helpful and important feedback that informed the design of the program. So as I already mentioned, we've made the decision to limit the number of biopsies. We also have done an open label study, understanding that being on placebo is a very big commitment for the patients. So in this study, we know that objectively seeing dystrophin is evidence that N531 is having the effect that we hope it to have. We introduced a clinical trial concierge. We really understand that involvement in clinical studies is a huge burden for boys and their families, going to and from the site, being at the site for the long days, assessment. So we've done what we can to try and minimize that burden for our boys and their families, and minimize the out-of-pocket expenses that they experience during this, and introduction, where it's possible, of option home nursing, and really thinking about how we can best communicate our results. We're grateful for this opportunity today to talk about this interim result from FORWARD-53, and fully committed to continue to provide insight to this as we move through the study. Now, of course, exon 53 is just one exon, and although there's potential for this to benefit, boys with exon 53 amenable skipping, the question for others, of course, is when will you be getting to the other exons, and will the exon that's relevant for me be subject to your studies? So we're really excited about the results that we have seen in this interim result. We're in the process of engaging with regulators on understanding the pathway forward for accelerated approval and expanding into other exons. So you can see here on the right, 51, 44, 52, and 45. And I think, with these other exons, we really have the potential to be addressing a significant population of DMD, people with DMD. It's never enough until every person has been addressed by an effective therapy, and we're really conscious of that. But we will really be moving forwards as quickly as we can to try and further expand with these positive results. So as a final thought before we open to discuss some of the questions, I think we really recognise that none of this is possible without the boys and their families, the clinicians and the study site staff participating. We're at the halfway point of the study. We're looking forward to completing the study in Q1 of next year. And we're excited to complete and further share results with you when they are available. Thank you so much.
Thank you.
Thanks, Paul. Thanks, Anne-Marie. So we had some questions come in. I think actually if we can go back and maybe start at slide 4, can clarify some different things throughout the talk. And then as those questions that came in, I'll kind of try to weave them in, make sure we get to everyone. So Paul, you did I think a good job of explaining this, but I think it's always helpful with the families who may be newer to the space, not as used to seeing some of the preclinical data. So with the mouse data, as you said, it's a different sequence for the exon skipper, but your backbone chemistry that you're using in your current product is what's remaining the same here, correct?
Correct. And so, no, great point. So it is the chemistry that stays consistent. And I think that's a really important concept, because what we do see in terms of the distribution, so as Anne-Marie showed in the translation of the clinic, really high levels of concentration of drug delivery into the muscle without the need for conjugates to deliver it into the cell. And so that's the same backbone that's consistent again across what we've gone into the non-human primate study, where we can measure that, safety studies, and then ultimately in the recent clinical data. I should say also from a safety perspective, we're using this chemistry in a backbone in a number of different programs in different cell types. And so we do see this consistent response. So it's not unique just in this circumstance.
And then the other piece of this that I think is important for families, just because they see a number of different animal models, and in Duchenne, we do have a lot of them. We have mouse, rat, dog, a whole host, but the non-human primate is not an actual Duchenne model. It's a little bit more similar to almost doing it in a healthy volunteer, right? So you just kind of explain that.
Yeah, no, thank you. I think we look at and I always say models are important for what you design them to do. And I think, ultimately it's to give confidence and conviction in going into the clinic. And I think to that point, as you said, the non-human primate model is essentially a healthy muscle. And so it was encouraging to see actually high levels of delivery into intact muscle cells. Oftentimes there's this discussion of maybe you need leakier cells in order to get more drug into them. So it actually gave us a lot more conviction that we saw really nice distribution into important muscle cells in that model. I think going back to the double knockout was important to us in evaluating that model, because we had done a lot of previous work on our prior DMD programs in MDX models. And I think our learning in that transition and the translation of the double knockout model, which was important to us, is that this model has a rapid mortality because it actually is an exacerbated phenotype of DMD, right? There's no dystrophin in any of the tissues. And so any advantage that you see in that model is solely coming from the production of functional dystrophin. And so that's important, not just in being able to observe the ambulatory patterns in these mice, but we can also do functional assessments. And I think what was the most encouraging data to come out of this was really the fact that respiration, we looked at functional tidal volume, how the mice were able to breathe. actually got back to wild type levels of respiration function. So it gave us a high degree of correlation that, we were producing functional dystrophin protein in its ability to translate.
Great, thank you. That's helpful. I think it's always tough when we're looking at the preclinical models, and especially with the challenges we've had with translation over the years, and it can be a lot for families who maybe aren't as steeped in it. So appreciate that. If we go to slide 6, Emery, you talked about this being potentially registrational. And again, just hoping you can talk through what that means a little bit more for families who maybe are newer to the space, not used to some of kind of this language as we're talking about kind of regulatory pathways.
Absolutely. So I think there's a lot of regulatory precedent in this area of DMD of companies who have been able to use dystrophin as a biomarker that's reasonably likely to predict a clinical outcome. And so with this design, we're following a very well-trodden path of providing a body of data that would enable FDA to, in future, approve the drug and bring it to the market. But of course, one always has to be circumspect until approved, it's not approved. So we believe that this study provides the data that will allow us to have the right conversations with regulators and bring the drug to patients in the future. And we're engaging with regulators and completing this study, and the outcome of all that will be available in Q1 2025.
Great. Thank you. And then on slide 8, you talked a little bit about some of the mild drug-related effect. Or adverse event, could you just talk through what those were?
Absolutely, yeah. So there was a patient who had a platelet decrease, which was later repeated, and the patient actually was above the upper limit of normal. There was a mild pruritus, so a patient who had itching, and there was a viral vomiting. So those were the reported events. Nothing from our point of view that was worrisome.
And then on slide 9, we had a few questions come in. So specifically talking around the isoform kind of question, which I think is maybe some families more familiar with that than others. Could you talk through when you're kind of discussing these two isoforms that are consistent with Becker, what you mean by that?
So this is normal in the way that these drugs work. So the drugs help restore the reading frame, and this will enable the RNA to be formed. And there are multiple translational start sites and splicing that can occur. And in Becker, this actually results in many different isoforms of dystrophin. And those different isoforms support, luckily for patients with the Becker phenotype, a much milder disease course. And so we are seeing truncated dystrophin, which is consistent with Becker.
And I think, yeah, it's helpful for families is there's patients with different variants and deletions can still be amenable to these exon skipping therapies. So you're going to get different forms of the dystrophin kind of produced depending on what your particular variant is. And then also, so we've been seeing this more recently as companies have been looking at quantifying kind of dystrophin expression, is this discussion around unadjusted dystrophin expression and then, adjusted for muscle content. And I think it's helpful kind of in these kind of points to maybe talk through a little bit what that is, kind of what the difference between those two numbers are and how are you getting to that muscle adjusted kind of content.
Yeah. So when the biopsy is undertaken, the surgeon chooses an area of the muscle that visually, looks like a reasonable area to biopsy. And then the biopsy is taken to the lab and it's sliced into very thin slices, and quality checks are done and it's analyzed. And of course, there will be a huge amount of variation in the actual site that's been analyzed. And so when we do the muscle content adjusted we're basically taking into account the amount of fibrotic and fatty tissue that is in the muscle sample that's being analysed at that time. And so we feel that this actually perhaps provides a truer reflection of the amount of dystrophin that's being produced, because the fibrotic and fatty tissue is not able to produce dystrophin. So it's basically a correction for the amount of fibrotic and fatty tissue that was available in the sample that was analysed.
Great. Thank you. It's helpful. I think it's anytime we kind of new terminology coming in, it's helpful to kind of repeat every time we see it. And then for these, were they open or needle biopsies?
These were open biopsies, and again, we'd really like to thank the boys for the contributions they've made here. We do understand that this is significant contribution by the boys.
And then on slide 12, I think you did a good job of explaining this, but just for in case families missed this. So the patient 1, part A, that is after 3 doses, I'd assume.
3 doses and 6 weeks of follow-up. And then on the right, this is the patient through essentially 6 months of treatment. This is how the tissue looked through 6 months of treatment. Now, of course, they're different samples, but the same boy. So, I think a good way of thinking about what's occurred in the 6-month period. And so, as I mentioned, the fact you can see more of the black arrows, these are the internalized nuclei. So that means the cells are becoming healthier. And similarly, on the bottom row, you can see the change in sort of the formation. They're just more regular and round. And this is also an indicator that the muscle health is improving.
Great. And then on slide 13, so you showed some of the biomarker responses, and I just wanted to clarify, is this the mean for all of the patients in the study, the 11 boys?
That's right, yeah. And CK is a somewhat noisy biomarker. We're very conscious of that, but I think what was really striking for us is this overall decrease that we're seeing, and also the fact it's correlating with AST. So, in the context of seeing the dystrophin, this is really encouraging.
And then on the next slide, so we had a few questions kind of come around a few aspects here. So one of which was you talked both in the preclinical data and then kind of hope that N531 is getting into the heart and the diaphragm. And so one of the questions we had was around plans for maybe looking at expression of the dystrophin in the heart and the diaphragm, which we can talk a little bit about the challenges there. And then aside from that, are there outcome measures that you're collecting that are going to maybe help to give us some insight?
Thanks. That's a really great question. So the way that we determine the dystrophin in the skeletal muscle is to do a biopsy, and we wouldn't do a biopsy of heart or diaphragm because that would be too dangerous for patients and not at all ethical. So we can't measure directly dystrophin in boys in the heart and diaphragm, and so any inference that we have will be taken from clinical outcomes. So in the study, we are looking at cardiac echo and MRI. These patients are thankfully in good health with respect to their respiratory and heart. And so we wouldn't necessarily expect to see much change in the course of this study, but we are measuring things like also the ability to expel air from the lungs, a measurement called FVC, forced vital capacity. And I think thinking about the future studies that we need to conduct, we will be thinking about more specific studies just to try and tease out what the impact of getting dystrophin into heart and lungs could be. And I think, it's too early to say what those studies would be, but that's something we're really deeply examining and thinking about the designs going forwards.
And then similarly on some of kind of future studies, there were questions around would you be looking at any additional or further dose escalation? And I know you talked already about based on the half-life, that you're able to move to a 1-month dose rather than kind of the 2 weeks. So just if you could address that question.
Oh, so what these results have shown us is that we're definitely in the right therapeutic range for the dose. There's absolutely plenty of drug in the tissues and the cells, and there's really no for us to be escalating in dose further. We're really in kind of the sweet spot. And the fact that we have so much drug in the tissue and we have the 61-day half-life, as you mentioned, gives us the ability to dose less frequently while maintaining enough tissue, drug in the tissue that will enable it to continue to support sort of dystrophin expression we're already seeing. So, there is actually so much drug that perhaps even less frequent might be possible in future, but we feel that the monthly dosing is really going to optimally support limiting the impact and burden on boys in terms of drug administration and also supporting dystrophin expression in future.
Great. And then we had a question around, as you are talking and thinking about kind of your regulatory path in kind of the post-approval landscape. We have seen at times challenges with some of the payer policies for getting access to some of our non-ambulatory patients who maybe weren't included in the original clinical trials, because payers can sometimes push back and they'll say that, we don't have data from the trial to support, which, obviously then our clinicians have to work to argue with them to get access for their patients. Do you have plans on expanding and your cohorts in future studies to ensure that some of these non-ambulatory patients, there's data collected, whether it's safety or some efficacy data?
Yeah, so I think this is a really important question. It's one of the reasons why we did open the study also to non-ambulatory boys. And obviously we only have 1 out of the 11, but I think this is a really important start. And when you think about our results, the one non-ambulatory patient who was on the right of the bar, this patient does have a lower response than the others, but is still achieving more than 1% dystrophin in the skeletal muscle, which is, more than some market or around the same level as some marketed products at the moment. So this is still an important result for non-ambulatory patients. Also adding in the point that you made about, the potential for the heart and the diaphragm for the dystrophin that could be being created there and the impact that could have on non-ambulatory patients. So as I mentioned, we're thinking about how we can be further understanding that impact in heart and diaphragm, and by definition these patients are more likely to be non-ambulatory patients because their disease has unfortunately progressed further. So that's certainly within the concept of the studies that we'll be thinking about in future. Oh, and I should just say this non-ambulatory patient, the biopsy was undertaken in the bicep of this patient.
And then we had a similar question kind of on the opposite end of the spectrum of plans to go younger. And I think this is especially relevant as we have a few states that have begun newborn screening for Duchenne. So starting to, potentially identify newborns. And if we have approved products, I think we want to, be able to initiate interventions as early as possible.
Yes, that's certainly something we would want to and need to do, of course, because all sponsors are required to think about the whole pediatric population. And the advent of newborn screening actually is a wonderful step forward for the community and something that we fully support. And we absolutely are open to involving younger participants as we move the program forward.
I think just to add to that, I think the work that we've done in terms of accessing the stem cells uniquely, right? So the regenerative cells in muscle, so beyond producing dystrophin, but getting to those restorative cells early before muscles become fibrotic. And it does, to Anne-Marie's point, create a really unique opportunity.
Great. And we also had some questions kind of on, I think, potentially future sites and I think families that are maybe wondering about eligibility of will they have opportunities with either US sites open or sites in Europe as you're thinking about future studies?
Yeah, I can't announce today any specific countries, but clearly one of the future steps for this program will be a confirmatory study, and those studies are normally large and would require us to open sites in many countries and sites, multiple sites in many countries. So I would foresee there being many opportunities for participation of this community for further developing N531.
And then I think the last question we had, maybe in the next slide, I think, just to cover again, or maybe it's one after this, but essentially there's a family asking kind of what is kind of you're considering next in the pipeline, which you touched on a little bit here, but I think families see data, they're encouraged, but they look and they see their mutation maybe on the on the map, but not a lot of clarity as to maybe when or how they'll get there. So I don't know if there's any additional input you can provide, or I think one of the things PPMD is hopeful for, and we've made comments on a recent FDA guidance that there will be some flexibility with regulators to speed up this process, so that way you all don't have to start from scratch every time we go through this, but kind of build on some of the evidence you've generated for some of these other exons.
I'm so glad you mentioned that, Eric. I think it's a great point, and I really commend PPMD for the work you've done to support that. So what we're talking about here in concept is, first, that if you are developing drugs in a number of different exons, there's a lot of similarities between those drugs. And so there should be ways from a regulatory point of view to leverage the data you have across all of these different molecules in terms of how you manufacture and the safety that's coming from animal studies and human studies. So that's because we have these plans with the different exons is absolutely something we'll be exploring with the regulators. And the second point is the time it takes to develop these drugs is also very much bound in the time it takes for us to execute the clinical studies. And so there's a very unique opportunity here to do something that's called a platform study. What this would involve is enrolling patients who have lots of different deletions, for the different exons like we have here. And then we could share a placebo arm for these different exons, and that would enable us to develop multiple new drugs in parallel and just make use of the placebo arm, which is such a significant sacrifice for the boys who sign up and then end up in this placebo arm, make that much more efficient and helpful for many of the different drugs rather than just one. So this concept of a platform study, I think, is going to be really transformational in the field of DMD, and it's something that we fully support, and hopefully will then bring these new therapies to the market much quicker for the boys and their families who are waiting.
Yeah, we definitely want to see that process sped up, so we can get to as many families as possible. I think with that, we are at the end of the questions in the chat. If you are watching this on playback and you have questions still, again, please reach out to us at PPMD. We will either answer your question or get you connected with the folks at WAVE to make sure that you can get a clear answer. But I'd like to thank both Paul and Anne-Marie for the presentation today, taking time to walk through all those questions and provide some really great explanations. I think that's really helpful as we kind of talk on a number of different topics within that. So thank you both.
Thank you, Eric.
Thank you.